Dear students, uh, in the last video, I had discussed with respect to the introduction to genomics, proteomics, and metabolomics. Uh, in that, we learned what do we mean by genomics, what does genomics actually consist of, and how this genome analysis is conducted. In this video, I'll be talking about the functional genomics, which is an important part of your syllabus. So, what do actually functional genomics means? Functional genomic means it's a part of molecular biology that attempts to describe a gene. In turn, the function of a gene in the form of a protein. So most importantly, in case of functional genomics, we will be actually analyzing the functional aspect of a gene either in the form of its functional RNA or the ultimate form in the form of its protein. Okay, so we will not only stop there, we also try to interconnect between the two components, how the genes will interact with each other. So functional genomics actually makes use of vast data which will be generated by analyzing the transcripts through transcriptome projects, which is the overall concept which is given in your syllabus. So functional genomics focuses on the dynamics aspects such as gene transcription, translation, regulation of gene expression, and their interaction with each other either in the form of RNAs or in the form of protein with protein. Okay, so usually the characteristic feature of functional genomics is to study the entire genome in a wider aspect. Usually how it was studied through high throughput methodologies which mainly included gene by gene approach. So but now it has changed. The goal of functional genomics has changed. So today it is mainly understood in the form of functional aspects. Okay, so this include either in the form of biochemical function, cellular, physiological aspect of each gene product is studied very carefully and analyzed. Through this, we will be able to assign the functions related to the genome itself, or it can be compared with other genomes also with respect to the mutations it holds or the polymorphism it displays. So through this we will be able to analyze the actual molecular activity of a genome through transcriptomes and proteomes, which also includes the metabolomes. So the functional genomics uses mostly multiplex techniques which measures the abundance of many or all gene products such as mRNAs or proteins within a biological sample. So for that what we do the RNA and protein assays. So assays to define the genome function. So what we do here, all the genes of organisms are usually not functional. In different group of organisms, the percentage of functional genes varies. For example, in bacteria, it's about 3 to 5 genes are non-functional, whereas in humans, 97% of the genes are non-functional. Besides, the level of evolutionary conservation of microbial protein is usually uniform with around 70% of the gene products. So that's why we try to connect the closely related bacteria through transcriptomes and the metabolomes. Each of the sequenced genomes has a homologous in distant genomes, homologicity in distant genomes. So we also try to analyze the comparative analysis of the genomes. Thus, the function of many of these genes can be predicted by comparing different genomes and by transferring the functional annotation of the proteins from the better studied organisms, which we call it as the model organisms, and compare them with the lesser studied organisms. So example like the Drosophila can be used as a model organisms as an entomological product to study all the different organisms related to entomological aspects. So in the same way, the C. elegans is used. In the same way, for plants, the Arabidopsis is used. Thus, it is possible to know by comparing different genomes and by transferring the functional annotations of proteins for better with respect to the better organisms, either like in the form to find the orthologous genes, the genes that are connected by vertical evolutionary descent, or same gene in different species, or paralogous genes, genes related to duplication within a genome from the lesser with compared with lesser studied organisms. So the functional genomics characterizes what sequences do what function. The goals of the functional genomics include the identification of all the RNAs ultimately transcribed from the genome, called the transcriptome of that genome, and all the proteins encoded by the genome, called the proteome. Functional genomics exploits both bioinformatics and laboratory procedures, which is based on the experimental designs. So ultimately, we have to note that through this functional genomics, we will be able to clearly find the function of the functional functionality of the genes. It can be 50%, 60%, 70%, whatever it is, whatever may be the percentage with respect to bacteria. In case of higher organisms, it may be around 3 to 5%. We'll find, but we will not stop there. We will try to use them, the organisms which is well studied, as the model organisms, and we will try to compare them with the lower studied organisms. Is it clear? So in the rest of the videos, we will be describing, we will be discussing how exactly we will be assaying and analyzing these aspects. Is it clear? Thank you.